Okay, so now we're going to go through a shift in the class. The previous lectures have focused on general virological principles, but now we're going to shift gears and start looking at specific virus families. This first lecture is going to be concerned with picornaviridae, caliciviridae, and hepaviridae. But first, before we get into the families, let's look at this overview of the replication of the genome in RNA viruses. So we're talking about transcription here. And we've talked about this a bit. Remember when we discussed the Baltimore classification? That was based on the genome of the viruses. Well, when we look at RNA viruses, we have three basic types of genomes that are possible. It can be a positive sense RNA, single-stranded, a negative sense RNA, also single-stranded, or a double-stranded RNA. Those are far fewer. There are a lot of positive sense and minus sense RNAs, but there are real substantial differences between a positive sense RNA virus and a negative sense. With a positive sense RNA, you need to remember positive sense means that it's the same sense as messenger RNA. So a positive sense RNA virus, as soon as it enters an infected cell and is uncoated, that RNA can be directly translated into viral proteins. And that will then be essential for carrying out the rest of the replication. Remember, for instance, that when you have an RNA virus, you're going to need an RNA-dependent RNA polymerase at some point. That's going to be a viral protein that has to be translated. When we look at the minus sense RNAs, they cannot be directly translated upon entering a host cell. So they have to carry a transcriptase in the virion. So that minus sense RNA will be transcribed into messenger RNA, which can then be translated. So that's a major difference between these two groups of viruses. So let's move on and look at some specific virus families. So the first three virus families we're going to talk about, the ones in this lecture, are all positive sense RNA viruses. Now with the family Picornaviridae, in terms of physical characteristics, they're naked, so there's no envelope. They're small, they're icosahedral in symmetry, only about 30 nanometers in diameter, and they have a single-stranded RNA that's positive sense. Now, the picornaviruses have been pivotal in the foundations of virology. Original research on ultrafiltrable infectious agents such as foot and mouth disease and poliovirus began the era of animal virology. Think back to one of those early lectures. Remember Leffler and Frosch and so on. The development of cell cultures for poliovirus replication led to Salk's inactivated and Sabin's attenuated vaccines. The first animal virus engineered into an infectious clone, and the first virus synthesized outside the cell was poliovirus. So these viruses have been widely used in the laboratory, and from a medical standpoint, they're extremely important. So if we look at their characteristics, naked, icosahedral, small, single-stranded RNA, positive sense. If we look at their classification, there's several genera in this family. They're not distinguishable by electron microscopy, so they all look very similar. They do differ in antigenic properties, also in pathogenesis, host range, and environmental stability. So let's look at some of the more important ones. So again, Picornaviridae is a large family of vertebrate viruses that produce both clinically asymptomatic infections, but often mild and even fatal disease. Their current classification includes more than 30 genera and 75 species, including several that infect humans. The genus Enterovirus comprises 7 species infecting humans. The genus contains poliovirus, Coxsackie virus, enterovirus, echovirus, and rhinovirus. Further serological classification results in hundreds of serotypes. Hepatitis A virus, or HAV, is the sole human virus species in the genus Hepatovirus. And they keep adding more genera. But if we look at this, aphthovirus has an important member, foot-and-mouth disease, which is an extremely important agricultural pest. It affects cattle and swine. Cardiovirus causes encephalomyocarditis. It's more important as a laboratory model. It infects mice. Now, enterovirus, that's a very large group. You can see 100 different serotypes. And we'll talk more about poliovirus in a bit. Coxsackie virus was discovered in Coxsackie, New York. Nowadays, when viruses are named, they're named after the place where they were first discovered. So Coxsackie virus was first discovered in Coxsackie, New York. And then the echoviruses, they're interesting because echo stands for enteric, cytopathic, human, orphan. Orphan means that when they were first discovered in cell culture, they weren't associated with any specific human disease. And we have the rhinoviruses. Those cause colds. They're not cross-protective, though. So we have many different serotypes. And that's why when you see, young kids, they always seem to be sniffling and snuffling, and that's because they'll have many colds a year, whereas adults usually just have one cold a year because we've been exposed and developed immunity to some of these serotypes, whereas a child hasn't. Another interesting thing about the rhinoviruses, and keep in mind they get their name rhino from nose because it's primarily a upper respiratory tract infection. Early attempts to isolate rhinoviruses were not successful. Standard cell culture conditions in a lab are 37 degrees C and 7.4 pH. However, rhinoviruses replicate in the upper respiratory tract. So the temperature there is going to be lower, and the pH is also going to be lower because of the high CO2 content of expired air. So when they changed the temperatures and started growing them at 33 degrees centigrade and at a lower pH, what they found is that the virus would replicate well. Now let's look more closely at the genome organization of these viruses. I've already said it's a single-stranded positive sense RNA. There's one open reading frame, so when this virus is translated, it's translated into one long polyprotein that is eventually cleaved into separate proteins. When we look at the genome, there's 3 regions. Region 1, you can see that in red, A, B, C, D, that encodes the structural proteins. Regions 2 and 3 encode proteins for RNA replication. You'll notice VPG attached. That's a small protein that's covalently bound to the 5' end, and it acts as the primer for initiation of RNA synthesis. The 3' end of the RNA is polyadenylated, meaning that it has many adenine residues attached. The track is heterogeneous in size, but mean size is genetically determined. Removing the tail makes the RNA non-infectious. So as an example of what I mean by heterogeneous in size but genetically determined, if we look at foot and mouth disease virus, it has a mean of 100 residues in this poly-A tail, whereas EMC virus only has a mean of 35. The function of this tail isn't known, but if you remove it with ribonucleases, it reduces the specific infectivity of the virus. If we look at the capsid, it's icosahedral, T equals 1. There are 4 proteins, VP1, 2, 3, and 4, that make up the capsid, and you have 60 copies of each VP protein per virion. And each one has a different function. So VP1, that's responsible for attachment to the host. So it's the anti-receptor. This is the protein on the capsid of the virus that determines the host range. They've been able to show this by doing some really simple experiments. So for instance, if you take just the enzyme trypsin, that will selectively cleave VP1. So if you treat the virus with trypsin, you find that it can't attach to susceptible cells. It'll lose its potency as a killed vaccine. It can't stimulate neutralizing antibodies. So all of these factors together were some early experimental evidence that VP1 is the anti-receptor and a major surface antigen. So, but keep in mind, as a naked virus, the capsid serves important roles. Protects the RNA from nucleases and so on. VP1, which is one of the capsid proteins, is the anti-receptor and major surface antigen. It also carries directions for selecting and packaging the viral genome, which is going to occur at the end stages of replication in an infected cell. But it also plays a role in penetrating the plasma membrane and delivering the RNA. So we'll look at these things in more detail in a minute. So remember, I talked about how important the replication scheme for viruses are, and we're going to be looking at the replication cycle of each of the viruses that we consider throughout the rest of this semester. And I can't stress enough the importance of this. Think back to when we were talking about antivirals and vaccines and so on. You have to understand the molecular replication of a virus in order to be able to attack it in an effective way to develop things like antivirals. Also, knowing the details about the molecular replication will give us insights into pathogenesis, host range, and so on. So we're going to go through the different steps of replication for each of the virus families that we're going to talk about through the rest of the semester. So let's look at attachment first. So with the picornaviruses, electrostatic attraction is an important component of attachment. If you just change the pH in the medium, that can change the attachment affinities. There are specific receptors on the cell surface that are necessary, and these explain, at least in part, the cellular tropisms that we see for the viruses. Now remember, the receptor is produced by the cell. It's regenerated, we know that, but we don't know a lot about its structure or function. but we do know that the receptor must have a function other than virus attachment. The virus must adapt to the cellular receptors and not the other way around. So in this simple experiment that's illustrated in this table, we took 3 different viruses in this group: poliovirus, Coxsackie virus, and rhinovirus. And what they did as a first step, if you look in the first column, you can see that they saturated the receptors. So for instance, look at polio. They took their cells, they put an abundance of poliovirus into the cell culture, into the cell culture flask, allowed time for attachment. And then they tried to challenge. And what they found is that if you used a different type of poliovirus, the first infection with polio would interfere with that. That's what the plus means. But notice the minus for Coxsackie and rhinovirus. The saturation of the receptors with poliovirus did not interfere with Coxsackie or rhinovirus. Do the same experiment with Coxsackie, so you saturate the receptors with Coxsackie virus, and poliovirus is not affected, rhinovirus is not affected. And a similar pattern with rhinovirus. What that tells you is that each one of these viruses is using a different receptor. So the next step is penetration and encoding. Interaction with the receptor leads to major structural changes in the virus. The poliovirus is a non-envelope virus, and it uses 2 alternative strategies to enter the cell. The virus can be endocytosed, which means the hydrophobic part of the viral protein VP1 will insert inside the cell membrane of the endocytic vesicles and form a pore to allow the penetration of the viral RNA. In the second strategy, the virus attaches to the cell membrane and directly releases its RNA into the cell. But in either case, VP1 changes conformation and forms a pore in the cell membrane. So it's this interaction with the receptor on the host cell membrane that leads to major structural changes in the virus, and that allows for the RNA to be released into the cell. So that's the uncoding. Then we move on to translation. Remember, that's going to be the next step. Once you have uncoded the RNA and it's entered the infected cell, because it's a positive sense RNA, it can be immediately translated. Now, we find that there's only one open reading frame with the genome for picornaviruses, and it's translated as a single polyprotein. And that initial polyprotein will then undergo cleavage, first an autocleavage, and then enzymes will carry out further cleavages. And that initial long polyprotein gets cleaved first into 3 parts, P1, P2, and P3, and then each one of those 3 gets cleaved into further proteins. You can see that P1 is associated with the structural proteins. The capsid proteins are a result of the cleavage of that first P1 precapsid protein. But the thing to keep in mind is that this means that all of the proteins are made in equimolar amounts. This is an inefficient way to replicate. Think about it for a minute. You only need 1 VPG. Remember, that's the protein that anneals to the RNA genome, or you only need 1 copy of replicase because it's reused, but you need 60 copies of each capsid protein. So essentially, you're making 5 million unnecessary replicase molecules to generate enough capsid for 1 million progeny. Again, like I said, it's a very inefficient process. After the initial rounds of translation, the RNA must disengage from the ribosome, and it will serve as a template for RNA transcription. So that's going to be the next step for this. But let's look at a pulse chase experiment. Remember, we saw this slide earlier. So across the top, you just have time. And remember, large proteins are going to stay at the top of the gel. Smaller proteins are going to move down the gel. So they did a pulse chase experiment. They labeled, just a brief labeling, and you can see that at the top of the gel, in the early stages of this infection, you just had 1 band, a very large band at the top of the gel. But over time, that large band got smaller, and you started to see bands moving down the gel, meaning that those bands were proteins that were smaller. And eventually, at the end, you've got a lot of individual small protein bands, and the large band that you started out with disappeared. That was the first way that they demonstrated that with picornaviruses, the translation was 1 large polyprotein that was then cleaved into smaller proteins. Okay, so that's translation. Now we want to move into transcription. Eventually, you have to move into transcription because you have to make copies of the genome in order to have infectious virus. So you can't just translate proteins. You have to eventually transcribe RNA. So what we see is early in infection, mostly we have the viral RNA acting as messenger. Then in the mid stages of replication, as more replicase is available, we shift into making minus RNA. So you move into minus RNA transcription. And that's an essential step because remember, if you're going to make more positive RNA, you're going to have to go through a minus RNA intermediate through base pairing. That's the only way that you can replicate RNA and DNA. So you have to eventually shift to making a minus sense RNA, and that's what you see here. So at the top of this slide, you see virion genomic RNA. After a period of time when viral proteins had been translated, you eventually had enough replicase, because replicase is one of those proteins that's going to be translated. Because remember, it's an RNA-dependent RNA polymerase, or a replicase. Eukaryotic cells don't make those. The virus has to encode it. So eventually, you have enough replicase that you start making minus sense RNA. And that's what you see in the middle of this slide. The purple lines are the opposite sense RNA. So you started out with positive. Those are negative sense RNAs that are being transcribed. And then eventually, those minus sense RNAs will act as template for more positive sense RNA. So when you think about that input RNA, it's positive sense, there are 3 possible fates for positive sense RNA inside an infected cell. It can act as messenger RNA. That's going to be the primary thing that's going on early in the infection of the cell. Ultimately, as you build up more replicase, it can act as template for minus sense RNA. That's an important intermediate step to make more positive RNA. And then eventually you move into making genome for new virions. Okay, so now we're ready to move into assembly. The protomers self-assemble into pentamers. So go back and look at what we talked about with icosahedral symmetry. And you have the VPO, VP3, and VP1 are the three capsid proteins. They're going to assemble into protomers. That's what you see in this illustration. And then those protomers are going to assemble into pentamers, or groups of 5 protomers. And then those will assemble into the procapsid. It's only after the RNA is encapsulated that the protein VPO is cleaved and the infectious virion is complete. So that's what you see here. So first you make a procapsid from assembling all of your pentamers, and then after the RNA, the genome of the virus, is inserted into the procapsid, you get cleavage of VPO into VP2 and VP4, and now you have your complete infectious virion. So a question would be, why the late cleavage? Why do you have VPO that's then cleaved into VP2 and VP4? Well, it's probably to change the structure of the capsid. Because remember, you have to get the RNA inside that capsid first, and then you have to lock down that capsid so that it can protect the RNA. So the procapsid, before VPO has been cleaved, is not as tight a structure. So the RNA can be inserted, and then once the RNA is in there, you have that final cleavage, and the virion is locked down. And now it's going to be infectious, and it's also going to be resistant to a lot of environmental factors. Some of these viruses, in fact, most of them, are going to be going through the digestive tract, so that RNA has to be protected. Okay, so here we have an overview of the coronavirus life cycle. So this is putting all of these pieces together and then talking about how it exits. So after binding to its receptor and endocytic uptake in the cell, the virion delivers its positive strand RNA genome across the endosomal membrane into the cytoplasm. The viral genome is covalently linked to the viral protein VPG, which is required as a primer for replication. Genome translation yields a single polyprotein that is proteolytically cleaved into replication proteins and capsid proteins. Genome replication by viral RNA-dependent RNA polymerase starts with synthesis of a negative strand RNA that serves as template for synthesis of new positive RNA molecules. Nascent positive RNA molecules can either enter a new round of replication or be packaged into progeny virions. Capsid proteins self-organize into protomers and pentamers, and in concert with replication machinery and genomic RNA, assemble into provirions that are converted into infectious mature virions upon the genome-induced cleavage of VPO into VP4 and VP2. Mature virions exit the host cell either by non-lytic release of extracellular vesicles or by cell lysis in a late stage of infection. So you can see in this diagram the 2 types of release are possible. But this diagram just summarizes everything I just talked about in these previous slides. So this is, putting all the different steps together. Next, we want to talk about how the virus infects its host cell. Now remember, the susceptible host cell is going to contribute energy. It's contributing all the precursors that the virus needs to replicate. So all the amino acids and some of the enzymes and so on, the ribosomes, everything that's needed for translation and replication. It also provides the receptors for infection and the membranes, the internal membranes that play a role in the replication of the virus. So that's what the host cell is contributing to this whole infectious process. Now, the virus is not a very good guest. It comes in and it inhibits cellular RNA synthesis and protein synthesis. Now, here's an interesting picture looking at the effect of picornavirus infection on RNA synthesis. And what we did in this slide here was just treat the cells with tritiated uridine. Now think about what kind of molecule takes up uridine. Remember, it's not DNA, right? Hint, hint, it's RNA. So you treat the cells with tritiated uridine. If you look at the cells at A, these have almost all of the RNA in the cytoplasm and not very much activity in the nucleus. You can see that the browned areas in the middle of each of these cells is much less dark. That's because you've got pretty much a complete shutdown of RNA synthesis in the nucleus. But if you look at the left side, B, those are normal uninfected cells. Now look at and notice where RNA synthesis normally occurs in an uninfected cell. Remember, RNA is synthesized from DNA. Where's the DNA? In the nucleus. So this is a nice example of how the virus comes in and shuts down RNA synthesis by the cell, the host cell, in favor of its own RNA synthesis, which is occurring in the cytoplasm, not the nucleus. Okay, here in this slide you can see that there's a shutdown of cellular protein synthesis as well. So we have inhibition of cellular translation. And what's going on is you're getting an inhibition of the 5' end dependent messenger RNA translation. And this is occurring through the cleavage of an elongation factor, EIF4G. So if you look at that little graph on the left-hand side, you can see hours post-infection, rate of protein synthesis. With uninfected cells, the line climbs slightly. But with the polio-infected cells, you can see that the rate of protein synthesis drops off precipitously, and then it starts to climb back up again before it drops off again. Well, what happens is that first drop-off is a drop-off in cellular translation. The rate of cellular protein synthesis is going down. That second hump there is from poliovirus translation. So you're getting synthesis of virus proteins and not cellular. And you can see that too in this gel on the right-hand side. So across the top, 0, 1, 3, those are just hours. And so the cells were infected with poliovirus. And initially, at 0 and 1, you just have a smear because there's so many different viral proteins that are being produced. But then you look at 3, and you're starting to see fewer and fewer cellular proteins, and certain bands start to emerge. And eventually, particularly at 5, the bands that you see are of viral proteins and not cellular. And then ultimately the cell is near death, and you get a fading of the viral proteins as well. But what's happening is that the virus cleaves the cellular initiation factor, EIF4G. That shuts off cellular protein synthesis. And remember that the viral RNA has VPG as its cap, not the typical methylated cap. So it doesn't use that elongation factor to initiate its protein synthesis. So it can attack that that elongation factor and shut off cellular protein synthesis while allowing its own to progress. Okay, so let's now look at some of the diseases caused by members of the Picornaviridae. First, of course, is polio. This disease is characterized by death of motor neurons in the spinal cord. Most infections are inapparent or perhaps mild febrile illnesses, but in about 2% of cases it spreads to the spinal cord and central nervous system, resulting in paralysis. You can even get fatalities from respiratory muscle paralysis. If you think about the name poliomyelitis, which is used as a term for polio, polio means gray, and myelo refers to spinal cord. Now, in the top left figure, you see a cross-section through a spinal cord, and you see an illustration down below as well. In the spinal cord, you have gray matter that's composed of cell bodies that forms that sort of a butterfly form in the middle of the spinal cord. You have neurons with large cell bodies in the anterior horns, so that like the wings of the butterflies would be the horns. And the anterior horns, the fatter horns, give rise to motor fibers that pass out through spinal nerves to skeletal muscles. Most of the neurons in gray matter are interneurons. White matter, which is the rest of the spinal cord, consists of myelinated nerve fibers comprising nerve tracts. Now you can see in that cross-section of the nerve cord there's some arrows. Those are indicating some darkly stained areas. Those are indications of damage to nerves within the gray matter of the spinal cord. Epidemics in the 20th century occurred because of increased standards of hygiene, which, you would think that that would be a good thing. But what happened is by increasing standards of hygiene, particularly improved sewage treatment and so on, you delayed the time of infection. Before these improvements were in place, most people were infected as infants, and they were protected by maternal antibody and less likely to develop paralysis. But changes in human behavior brought about serious complications. And we'll see this again and again. Changes in human behavior can bring about serious complications related to infectious disease. In this case, we're seeing it in reference to polio. In the 1950s, there were more than 10,000 cases every summer, with about 40% developing paralytic disease. Many paralyzed patients were ultimately able to resume almost normal activities. Many learned to walk again, and so on. But what we found is that now, all these years later, as these polio survivors from the 1940s and 1950s are now in their 60s and 70s and 80s, we're seeing a situation called post-polio syndrome. This first shows up 30 to 40 years after the initial polio infection. It's characterized by fatigue, pain, and weakness. And we don't know exactly why it happens, but it could be from overuse of the surviving motor neurons. So here, if we look at this, post-polio syndrome, again, it's a condition that affects polio survivors years after their recovery from an initial acute attack of the poliomyelitis virus. Most often, polio survivors start to experience gradual new weakening in muscles that were previously affected by the polio infection. The most common symptoms include slow, progressive muscle weakness, fatigue, both generalized and muscular, and a gradual decrease in the size of muscles, that is, muscle atrophy. Pain from joint degeneration and increasing skeletal deformities, such as scoliosis, which is curvature of the spine, is common and may precede the weakness and muscle atrophy. Some individuals experience only minor symptoms, while some develop visible muscle weakness and atrophy. Fortunately, post-polio syndrome is rarely life-threatening, but the symptoms can significantly interfere with the individual's ability to function independently. Respiratory muscle weakness, for instance, can result in trouble with proper breathing, affecting daytime functions and sleep. Weakness in the muscles you use to swallow can result in aspiration of food and liquids into the lungs and lead to pneumonia. There are several theories as to what causes post-polio syndrome, but no one knows for sure. But what they think is happening is when poliovirus infects your body, it affects nerve cells that are called motor neurons, particularly those in your spinal cord, that carry messages as electrical impulses between your brain and your muscles. A polio infection damages or destroys many of these motor neurons. To compensate for the resulting neuron shortage, the remaining neurons sprout new fibers, and the surviving motor units enlarge. This promotes recovery of the use of your muscles, but it also pushes the nerve cell body to nourish the additional fibers. Over the years, this stress may be more than the neurons can handle, leading to the gradual deterioration of the sprouted fibers and eventually of the neuron itself, leading to a return of the kinds of symptoms that the patient had early on, including potential limited paralysis. Now, within the group of enteroviruses, we have non-polio members as well. Non-polio enteroviruses are very common. They are second only to the common cold viruses, the rhinoviruses, as the most common viral infectious agents in humans. The enteroviruses cause an estimated 10 to 15 million or more symptomatic infections a year just in the United States. Everyone is at risk of infection. Infants, children, and adolescents are more likely to be susceptible to infection and illness from these infections because they're less likely to have antibodies and be immune from previous exposure to them. But adults can also become infected and ill if they do not have immunity to a specific enterovirus. Most people who are infected with an enterovirus have no disease at all, so asymptomatic. Infected persons who become ill usually develop either mild upper respiratory symptoms. We sometimes think it's a summer cold, a flu-like illness with fever and muscle aches, or an illness with rash. Less commonly, some persons have septic or viral meningitis. Rarely, a person may develop an illness that affects the heart, or myocarditis, or the brain, encephalitis, or it can cause paralysis. Enterovirus infections are suspected to play a role in the development of juvenile onset diabetes or sugar diabetes. Newborns infected with enterovirus may rarely develop severe illness and die from the infection. Now, as an indication of why they think that enteroviruses may be playing a role in diabetes, they found signs of enterovirus in pancreatic tissue from 60% of children with type 1 diabetes, but they rarely find it in children without diabetes. They also found that 40% of adults with type 2 diabetes had signs of the infection in insulin-promoting cells. The study published raises the possibility of a vaccine. Although genetics is known to play a role in a person's risk of developing diabetes, environmental factors must also be involved, and the idea of a viral cause of diabetes has been considered for decades. Hepatitis means inflammation of the liver. We will be talking about several different kinds of hepatitis viruses, all in different virus families, and they're all given a letter as an indicator. So in this case, we're talking about hepatitis A. Now, hepatitis A ranges in severity from a mild disease lasting several weeks to a severe disease lasting several months. It's estimated about 3,000 cases of hepatitis A occur in the United States each year, although only a smaller number of cases are actually reported to public health agencies because many infected people have no symptoms or mild symptoms and don't go to see a doctor. HAV can be found in the feces or stool of infected people, and their feces can contaminate surfaces, objects, food, or water. People get sick by swallowing the contaminated food or water, or by putting their hands in their mouths after touching contaminated surfaces or objects. The virus can also spread from person to person through sexual activities and sharing contaminated needles. Not everyone who is infected will have symptoms. If symptoms develop, they can include tiredness, poor appetite, fever, or nausea. Some people might also have vomiting or abdominal pain. They can also have a very dark urine. Jaundice, which is a yellowing of the skin and the whites of the eyes, might appear. You can see that in the picture in the lower left. Infants and young children tend to have very mild symptoms and are less likely to develop jaundice than are older children and adults. Symptoms usually appear within 4 weeks after exposure, with a range of 15 to 50 days. So if you look at the graph on the right-hand side, you can see weeks after exposure and the relative concentration of anti-hepatitis A antibodies. You can see that IgM is the first to develop with a peak at about 6 weeks after exposure. IgG peaks about 8 weeks after exposure and then continues on. But look at up above. You have virus in the blood first showing up at about 2 weeks after infection. Virus in the feces, and remember that's an important means of infection, starts to occur at about 2 weeks. Transaminases, those are just enzymes produced by the liver that are used as an indicator of some sort of liver infection. It's not an exact indicator of hepatitis A, but it's an indicator of hepatitis, disease of the liver. But notice the virus in the blood, the virus in the feces, those are 2 ways that the virus is transmitted. They show up a good 2 weeks before the actual symptoms occur, the symptoms such as jaundice and so on. So that contributes to the transmission of this virus. People who are largely asymptomatic are actually at their highest infectivity. When hearing about hepatitis A, many people think about contaminated food and water, and certainly, restaurants are sometimes closed because of an outbreak of hepatitis A. But in the United States, hepatitis A is more commonly spread from person to person. Since March 2017, CDC has been following viral hepatitis and looking at multi-state outbreaks of viral hepatitis, and these have been largely spread person to person. Hepatitis A incidence increased 850% from 2014 to 2018. The increase in 2018 was primarily due to unprecedented person-to-person outbreaks reported in 24 states among people who use drugs and people experiencing homelessness. Since the outbreaks were first identified in 2016, 30 states have publicly reported great increases in numbers of cases of hepatitis. So while hepatitis A is on the rise in the United States, it's been an ongoing issue in countries that suffer from high levels of poverty and individuals who do not have access to clean drinking water. The statistics for worldwide contraction of HAV is much more staggering. The World Health Organization estimated that in 2016, more than 7,000 people died from hepatitis A worldwide. One of the largest outbreaks of HAV was in 1988 in Shanghai, China, and it was related to food and water that affected more than 300,000 individuals. The Virginia Department of Health has identified an increase in hepatitis A cases statewide, consistent with the rise in cases in many other states around the country. If you look back at that map that we saw on the previous slide, you'll see that Ohio and West Virginia, for instance, have very high rates of hepatitis A. After a retrospective review of surveillance data, the Virginia Department of Health has determined that Virginia now meets the criteria to be considered an outbreak state and has declared a statewide outbreak of hepatitis A. But you can see on this map that the hardest hit areas are Southwest Virginia, particularly portions of Virginia that border West Virginia, which has been suffering high incidence of hepatitis A for a couple of years now. But look at the little graphic on the right-hand side, our smiling pile of shit. Sorry for the language, but that's what the emoji is called. It's telling you how to stop the spread of hepatitis. Wash your hands. But despite his big smile, the best way to prevent hepatitis A is to get vaccinated. So, the hepatitis A vaccine is the best protection against the disease. It's an inactivated vaccine, and it's available either as a 2-dose single vaccine just for hepatitis A, or you can get it as a combined vaccine with hepatitis B. Remember when we were talking about vaccines, we said that the hepatitis B vaccine was a recombinant. So Twinrix is a vaccine for both hepatitis A and hepatitis B. It combines two FDA-approved vaccines, the Havrix, which is an inactivated for hepatitis A, and the Ingerix B for hepatitis B. It protects individuals 18 years of age or older against both Hep A and Hep B. The vaccine is recommended for travelers whose occupation or behavior puts them at high risk for hepatitis B virus, or who are visiting countries with high or intermediate rates of both hepatitis viruses, as defined by the Centers for Disease Control and Prevention. So this lists the high-risk groups for hepatitis A, and of course, changing your behavior, practicing safe sex and so on, not using injection drugs, or at least making sure that you're using clean syringes. All of these things can help to prevent contracting hepatitis A. But the number one thing to do is to get the vaccine. I've had the Twinrix vaccine. It was recommended to me before I was making a trip to Kenya a few years ago. And I'm here to tell you, it's a simple vaccine. It doesn't really hurt. It doesn't make your arm hurt. It doesn't swell up. So, why not get it? The next family in this group we want to talk about is the family Caliciviridae. The name comes from the Latin word for cup or goblet, because they're cup-like depressions in the surface of the virion that you can see in the electron micrographs. The human caliciviruses have been difficult to study, so we know far less about them than the picornaviruses. But they are icosahedral. They're 38 nanometers. They have a positive sense single-stranded RNA with a 5' VPG and a 3' poly A tail. So they're similar in terms of genome, but they do have substantial differences from the picornaviruses. So first of all, they do have a larger diameter. They're a slightly larger virus than the picornaviruses. They have just one coat protein. Remember, the picornaviruses had 4. And there's a subgenomic RNA. We'll look at that in the next slide, but that's a substantial difference between the picornaviruses and the caliciviruses. Okay, so at the top you see the genomic RNA. That's the input RNA. And there are 3 open reading frames with this. So it's different than what we saw with the picornaviruses. Remember, the picornavirus just had one open reading frame, and a single large polyprotein was initially translated. With the Caliciviridae, we have 3 open reading frames, and so we have smaller polyproteins in the case of open reading frame 1 being produced, and then open reading frame 2 and 3 are associated with the virion proteins. Now, in addition, we have a subgenomic messenger RNA. So when we start moving into the transcription of RNA, we not only see the production of full-length genomic positive sense RNA, we also see a subgenomic messenger RNA. And that's what you see here. Now notice what's contained in that subgenomic RNA. It's the 2 structural proteins. Think back to what I was talking about with translation of the picornavirus proteins. I said that that system of using just one open reading frame that was translated into just one polyprotein that was then cleaved into all the individual viral proteins, I said that that was really inefficient. Well, the Caliciviridae have gotten around that by using a subgenomic messenger RNA. And it codes for just the structural proteins, which are ones that you're going to need in greater concentration than the non-structural proteins. So in this slide, we see the overview of the life cycle of a calicivirus. So after receptor-mediated endocytosis, the RNA is uncoated and translated in the cytoplasm through a VPG-mediated translation initiation mechanism that requires host cell factors. The non-structural viral proteins participate in the formation of a replication complex, the RC, and the RNA-dependent RNA polymerase, together with cellular factors that use genomic RNA as a template for the synthesis of minus-strand RNA. The negative strand is also used as a template for the production of new genomic positive sense RNA. Subgenomic RNA, which is also positive sense, is also produced and is further translated to generate capsid proteins for binding to viral genomic RNA. After genome and capsid assembly, the viral particles are released from the cell. So here you can see the virus enters, it's uncoated, but then notice, you go in and if you look towards the center of that slide where you see RNA replication, you're going to make full-length positive RNA, that's the gRNA that's indicated in the slide. And you can make more minus sense RNA from that full length, but eventually you can make subgenomic positive sense RNA. And you'll get eventually genome and capsid assembly and release of the particle. Norovirus is probably the most common and infamous member of this group. I would assume that probably most of you have gotten it, probably more than once because there's numbers of different kinds. But there's a limited amount of information on this because there's no cultured cell line for propagation and no animal model. The incubation period is short, 24 hours or less, and fortunately the course of the disease is also short, just 1 or 2 days. People often will get norovirus and mistakenly think they've gotten food poisoning, and that's not the case. Norovirus is the kind of virus that creates problems, for instance, on cruise ships and so on. The symptoms of norovirus illness usually include nausea, vomiting, diarrhea, and some muscle cramping. Sometimes people additionally have a low-grade fever, chills, and headaches, and just a general sense of tiredness. The illness often begins suddenly, and the infected person may feel very sick. In most people, the illness is self-limiting, with symptoms lasting for just 1 or 2 days. In general, kids experience more vomiting than adults, but it's important to make sure that whether it's an adult or a kid, they stay hydrated, especially if they're having serious vomiting and diarrhea. The illness caused by norovirus infection has several names. People will sometimes call it stomach flu, but I'm here to tell you it is not related to the flu. Do not call it stomach flu in front of me, because flu is a respiratory illness caused by influenza virus. So, this is different. This is a gastroenteritis, and you get an inflammation of the stomach and the intestines. It's also not food poisoning. Again, like I said, people sometimes think it is. But there are various ways that you can get it. Nursing homes are a common way, restaurants, vacation settings like cruise ships, you can see that in the settings, and mode of transmission. It can be foodborne, but it's not food poisoning in the sense that the food is not spoiled. There's no bacteria in the food or anything. You can get it person to person, waterborne, we don't really know. But no matter what, I'm here to tell you You don't want it. And then to finish, we want to consider family Hepaviridae. The spherical non-envelope virions of HEV are about 34 nanometers in diameter, and they resemble caliciviruses. Remember the cup shape to the calicivirus virions in the EM. These look a lot like that. They have a monopartite, linear, single-stranded, positive sense genome. The virions are icosahedral, non-enveloped, and again, like I said, a diameter of about 34 nanometers. The replication of these viruses is poorly characterized because there's a lack of efficient cell culture systems to study these viruses in. So we don't know as much about them as we would like, but it's thought that their replication process is similar to that of rubella viruses and the alpha viruses in the family Togaviridae. So rather than the other two viruses that we've talked about already, that they resemble superficially, it's thought that their replication is actually more similar to the Togaviridae. And if you look at these two illustrations, these trees on the right-hand side of the slide, you can see that the HEV genome closely resembles that of rubella virus, which is an envelope virus in the Togaviridae. It's conceivable that HEV evolved from rubella virus either by deletion of the envelope glycoprotein genes or by a recombination event. So if you look at this, the yellow circles indicate the Caliciviridae, the purple are the Picornaviridae, you've got the Togavirus in blue, and in green we have the Hepaviridae, and they're actually more closely related to the Togaviruses than they are to either calicivirus or picornavirus, and that's despite some physical similarities. But notice too, they have a single-stranded RNA, and they do have a poly A tail, but they have a 5' methylated cap and no VPG at the 5' terminus. So that's a major difference between this group of viruses and the previous two. But they all have they're all similar in the sense that they're single-stranded, positive sense RNA naked virions. Hepatitis E is prevalent in most developing countries, and especially in those countries with a hot climate. It's widespread in Southeast Asia, Northern and Central Africa, India, and Central America. It's spread mainly through fecal contamination of water supplies or food. Person-to-person transmission of this hepatitis virus is uncommon. Outbreaks of epidemic hepatitis E most commonly occur after heavy rainfalls and monsoons because this disrupts the water supplies. Domestic animals have been reported as a reservoir for hepatitis E virus, with some surveys showing infection rates exceeding 95% among domestic pigs. Improving sanitation is the most important preventive measure, and this consists of proper treatment and disposal of human waste, higher standards for public water supplies, improved personal hygiene procedures, and sanitary food preparation. Thus, preparation strategies, or I should say, the prevention strategies for this disease are similar to those of many others that plague developing nations, and they require large-scale international financing of water supply and water treatment projects. There's no FDA-approved vaccine for hepatitis E currently available in the United States, but in 2012, a recombinant vaccine was approved for use in China. Most people with Hep E recover completely. During outbreaks of this virus, the overall case fatality rate is about 1%. However, for pregnant women, hepatitis E can be a serious illness, with mortality reaching 10 and even as high as 30% if the woman contracts the virus in her third trimester of pregnancy. Hepatitis E can also be serious amongst persons with pre-existing chronic liver disease, and that can lead to death. And people who have received a solid organ transplant and are on immunosuppressive therapy can also suffer mortality from hepatitis E.